All right, so first up today is Dr. Tim Bruce. He's a postdoc in Dr. Ken Cain's Fish Health Lab at the University of Idaho. He earned his bachelor's degree at the University of Western Ontario, a master's degree in biology at Purdue University, Fort Wayne, and completed his PhD in fishery science at South Dakota State University. He was a research scientist for Prairie Aquatech, a company that manufactures plant-based protein ingredients for aquaculture, and the aquatic animal health specialist and fish pathologist with the Missouri Department of Conservation before starting in the CAIN lab in late 2017. His current research focuses on novel fish vaccine developments and strategies, investigating the health and nutrition of cultured burbot, and emerging bacterial fish pathogens in the Pacific Northwest. So with that, Tim, if you want to take it away. And Tim, just to point out, your slides are showing the notes too.
Yep, absolutely. Let me see how we can get that next up here. There we go. Now we're busy.
There you go. Yeah.
Yeah, perfect. All right. Well, thank you very much, Nick. Appreciate that. So again, my name is Tim Bruce. I'm a postdoc in Ken Cain's lab here at the University of Idaho. So today I'm going to be talking about a few different trials that we've done over the past little while here, evaluating a novel Flavobacterium psychrophilum live-attenuated vaccine that was developed here at UI. And we're going to look at discuss a couple trials here looking at the assessment of the cross-protective ability of this vaccine. So as I mentioned, I started here as a postdoctoral fellow in November 2017. We're located at University of Idaho. We're in Moscow, Idaho, so not too far from Spokane, Washington, in the Pacific Northwest here. Currently in Ken's lab, we have two postdocs, Dr. Jesse Ma and myself, two PhD students, Luke Oliver and Maureen Matua, that are working on burbot nutrition, physiology, and economics. And then also we have two master students, Brent Vuglar and Evan Jones. Evan's working on some oral vaccine development, and Brent Vuglar is working on a new rat project we have with the SOTA lab at UC Davis, looking at these emerging flavobacterial infections in the Pacific Northwest. So the research focus in the CAIN lab is focused on vaccine development, emerging salmonid pathogens, and also the health of new cultured fish species. And we've been doing a lot of work with burbot lately here, pushing that species into commercial aquaculture operations and looking at the health and nutrition for the fish, making sure it meets those needs. I'm currently working on a USDA EWD project right now, looking at burbot immunology and nutrition, looking at how plant-based protein products impact the gut microbiota, and looking at potential avenues for curbing some motile airborne septicemia that we do see in the species. So ongoing projects, more specifically, as I mentioned, we work quite a bit with the live-attenuated FP vaccine here. That's getting ready to commercialize. We're working with an industry partner right now, so it should hopefully be on the market soon. Very excited about that. We've been investigating the cross-protective potential for this vaccine against other FP strains, as well as novel and emerging Flavobacteria, family Flavobacteriaceae isolates. And we'll get to that in one of the papers I'll be discussing here. Working with bacterial viral co-infections, and then again, working with plant-based protein sources in some novel and upcoming species. So today for the presentation, I'm going to focus on a little bit of background on flavobacterial infections and the vaccine development here at UI. Look at cross-protection in one trial, the cross-protection with a variety of psychrophilum strains that were collected globally, as well as some domestic strains here from the US. And then discuss some new work that we've done looking at the cross-protection with family Flavobacteriaceae strains, a project that we worked on with Michigan State. And then finally wrap up and talk about some future and ongoing vaccine projects that we have in the lab here. So we're going to start out broadly here looking at the family Flavobacteriaceae. We, in the fish health realm, we commonly come across and run into Flavobacterium pulmonare with Pulmonaris disease, Branchiophyllum with BGD, bacterial gill disease, And then, of course, Flavobacterium psychrophilum, the causative agent of bacterial cold water disease, and rainbow trout fry syndrome. So these novel Flavobacteriaceae, family Flavobacteriaceae, we've been seeing much more of these reported in the literature in the last little while. They're routinely captured in both global and domestic diagnostic cases now. What's interesting with these bacteria is they form biofilms. They can be very hard to disinfect, and also that relates to their pathogenicity and ability to attach to the fish epithelium, of course, and entry. And then they often target the gills and external surfaces for pathogenicity. Within the family Flavobacteriaceae, aside from the Flavobacteria, we also have Chrysobacterium species. These also appear usually to be the yellow pigmented colonies. They show some strain differences, but they also have differences with respect to their growth dynamics, temperatures for incubation, etc. And Locke and Feisel have previously reported on many of the Chrysobacterium species and Flavobacterium species in the Great Lakes area. And they've also stated that they may have well over 100 different genres. So this is very, very diverse, these bacteria. So zoning in within the family Flavobacteriaceae with Flavobacterium psychrophilum, these are gram-negative bacteria, often show up yellow on your TYS plates in diagnostic cases, and they grow in cold temperatures and therefore impact fish and infect at those cooler temperatures too. These often have a sort of a hallmark loss with the degradation of the caudal peduncle, skin lesions, erratic swimming behavior as this bacteria likes to go into the vertebral column, and then also exophthalmia that you'll see too. Currently, for mitigation efforts here in the US, we use fluorphenicol and OTC via veterinary feed directives. That's in diagnostic collaboration with the veterinarian, of course, and a proper diagnostic results to guide us there. However, we've been seeing that there are resistant FT strains on the rise too. So we're going to have to expand that toolbox that we have here. With respect to the industry and the impact that this bug has on younger fish, is it can absolutely decimate production stocks as it moves through the system. And then any surviving fish that have encountered this in their early life stages can also have some long-term morphological issues that will, of course, impact the marketability of those fish stocks when they become older to market size. So looking at the live attenuated FP vaccine, as I mentioned, we are currently commercializing this with an industry partner right now. It's known as the B17 ILM vaccine. And we've seen a very strong systemic response in fish that are vaccinated that's conferred for long periods of time. This is an immersion-based vaccine, so it's a great option for larger production settings. And we've been investigating rainbow trout and coho salmon to date here, but for fish species, we're going to expand that. I'll talk about that near the end of the presentation today. But we've been looking at various FP isolates to see the cross protection, as well as those family Flavobacteriaceae strains, seeing what abilities this vaccine has to protect against a wide array of pathogens here. So the first trial that I'm going to discuss today has recently been published in JFD. And with that, I'd like to acknowledge our co-authors here, especially Chris and Tom at Michigan State University for contributing to this publication, and as well as the next one that we'll have on the list here too. So for trial A, we chose 9 different virulent strains of Flavobacterium psychrophilum from both global and domestic cases. So what we want to do is determine if that vaccine was, of course, cross protection, can confer a protective immune response to rainbow trout after exposure. We wanted to look at the vaccination period itself and, of course, assess whether or not the fish developed a strong serological response following vaccination. And then wanted to bring this to the bench and look at protective antigens that were recognized across the isolates with SDS page and Western blot. So with this, the working hypothesis was, of course, the vaccine would provide cross protection against a wide variety of these FP isolates. So for the vaccination itself, we started with 2.5 gram juvenile rainbow trout in our flow-through systems here at the CNR building at the University of Idaho. We used a standard immersion method of vaccination. We did a 3-minute exposure in this case. We chose a mock or sham vaccinated group with TYS meat culture media, so that those fish, of course, were not vaccinated. We did a booster at 2 weeks, and then also collected sera for titer analysis every 2 weeks leading up to the actual challenge trial itself. So every 2 weeks for a period of 8 weeks, we monitored and mapped those ELISA titers. So in order to make sure that we got the correct amount of injected the appropriate amount of bacteria, we ran virulence assessments first to make sure we could mark in the proper CFU per mil on these fish, and we dialed that in with our virulence assessments. And then we used approximately 10 to the 7 CFU per fish. That was about the sweet spot for mortality in this case. For the enumeration of these bacterial strains, when we bring them out, we use a drop plate methodology for enumerating, getting our CFUs per mil, and then transferring that to CFU per fish. In both the virulence trial and the post-vaccination challenge trial, we did a 28-day observation period in this case, and collected spleen and kidney tissue so that we could have, of course, monitor the ongoing infection during that time. With the data captured here, we used one-way ANOVA to analyze the antibody data as well as the CPM data too collected from this trial. So here's an overview of the strain selection that we used in this study. As you can see, a variety of isolates here captured from around the world, and a variety of different sequence types here that we use too. Where I want to draw your attention is the CSF 25993 strain. That's the parent strain to the B17 vaccine. That's a sequence type 10, along with the 79 strain that was isolated in Pennsylvania, and then the 9910A2 here. So just keep your eye on the 79, 9910A, and the CSF 259, those sequence type 10 groups as we move forward. Looking at results from the virulence trial here, we can see that we obtained a wide array of virulence of CPM here with a diverse virulence across these strains collected. Our standardized CSF 25993 strain fell in at about 60% mortality here. So we found that we had a good range for the trial. So everything that we were throwing at these fish in the post-vaccination challenge, we assumed they would be virulent given the virulence assessment here. We also did an injection of the B17 vaccine in this fish and also found no mortalities too as a control group. Here are the antibody titers over the 8-week period of time here. We have our control fish on the right and our vaccinated fish on the left. And as you can see, every 2 weeks of mapping those fish, that's a FP-specific IgM response via ELISA. You can see that our vaccinated group was developing a strong systemic response through that vaccination period. Looking at the challenge that immediately came after the 8-week vaccination period, these are our CPM findings here in the graphic. We have our control group on the left and our vaccinated fish on the right. And this is each strain next to each other. And as you go across the board, you can see a reduction in the CPM, significant reduction in each case with the vaccinated group. So some really nice findings across those strains. When we take another look at this or spin the mirror a little bit and look at the relative percent survival, we can see that our 259 parent strain here had a 70% RPS. And then again, the other ST10 sequence types had the highest RPS values also too, which was interesting. So it shows that the vaccine may be very interactive with that ST10 group. And then a couple clinical signs here showing ulcerations from the trial. And you can see some yellow coloration around that ulceration too. Those are fish collected from the trial period. So going into on the bench, we wanted to also take a look at the whole cell protein profiles from whole cell lysates here. So we ran these out on SBS page gel and separated them based on molecular weight of the proteins. On 1 and 2 are the vaccine strain, and then the parent strain here, the CSF 259. They look the same. And we can see very conserved regions here across the different FT isolates, which is interesting. So we took some hyperimmune sera from fish and then probed these whole cell lysates, the proteins in there. And we also found that to have these very reactive areas here, approximately 250 kilodaltons, 60 kilodaltons, and some low molecular weight too, therefore bolstering what we found in the in vivo work too, showing that some shared regions of antigenicity here. So moving on to a second trial that we just recently wrapped up with Tom and Chris at Michigan State this past year, we wanted to look at some of these novel family Flavobacteriaceae isolates and look again to see if this vaccine was cross-protective against them, evaluate the serological response following vaccination, and then bring this again to the bench and look at the Western blot profiles. So we used a very similar vaccination method here with immersion vaccination in approximately 3-gram rainbow trout again, and again had that 2-week booster with that vaccination period. Looking at the strain selection, we used 3 different isolates from Michigan that Tom and Chris had sent us. So we had S21, which was a Flavobacterium species. So this was a non-psychrophilum bacteria. And then we had S25 and T28, and they were Chrysiobacterium species too. And then of course included the CSF 25993 parent strain in the trial as well. You can see some of the morphological differences here on the growth too from the colonies too. And that can be a real challenge in diagnostic cases, making sure that you're honing in on the correct pathogen, of course, for species identification. So looking at the virulence and pathogen challenge, we ran 2 virulence studies on these strains. So we ran one at Michigan State. Chris and Tom ran that in a 1.5 gram rainbow trout, and then we followed up here in an 11.5 gram trout to get a appropriate range. So we did that ahead of time. I'll show the results in a moment here. We used approximately 10 to the 8 CFU per fish, and again, collected spleen and kidney tissue across the board just to make sure that we could confirm the ongoing infection there. So very similar to trial A. So we have the dosing here in virulence study A. That was the 1.5 gram fish at Michigan State, and then study B, that was the 11.5 gram trout here at U of I. As you can see the dosing here. And then looking at the CPM data that we obtained in the smaller fish, we did see a great deal of mortality. Those strains appear to be very virulent in those smaller fish sizes. But as we got to an 11.5 gram fish, we did see a reduction in the mortality too. We often see that with FP as it seems to impact those younger fish. And we also did use a few different fish strains here and some variables that weren't the same. But we at least got a dosing range for some mortality here that we could plan for the vaccination, post-vaccination challenge. So again, looking at the IgM specific titers here in that post-vaccination period, 8 weeks, we can see that the vaccinated fish did take off again and showed a strong systemic response across the board. So we were prepped and ready for that challenge post-vaccination here. And then looking at the CPM in the post-vaccination period with the strains. So we ran the S21, S25, and T28 novel Flavobacteriaceae on their own. And then we also mixed all 3 of those together in a mixture, kind of a cocktail, and then injected the fish with those 3 different family Flavobacteriaceae isolates. What we saw, I'll draw your attention here to the box. These are the control fish. So our control CSF 259 showed approximately 50% mortality, and then very low mortality in our vaccinated group. So again, a significant degree of protection conferred there. And also our mixed group with those 3 family Flavobacteriaceae isolates mixed together, we also saw a significant protection on that side too. So this equates to approximately 94% RPS with the 259 and about 85% with that mixed group. What we did see, this was about 7.5 gram fish, I believe, at the time of vaccination, or sorry, at the time of challenge, post-vaccination. We didn't see much in terms of mortality and couldn't tease out much of an effect here with the single family isolates, though, family Flavobacteriaceae isolates. So that's something that we're going to look into in the near future. So again, getting to the clinical signs here, Tom and Chris put together the graphic on your left here. You can see the ulcerations that we have here, some of the liver pallor here and some exophthalmia in the fish. Those were collected during the initial virulence. And then on the right, we have some of the fish that we collected during the challenge trial work here, the post-vaccination trial. You can see some of the yellow tint and colorations to these fish, some of the deep ulcerations near the sub-Q injections, and then we see some edema in these fish at the same time too, some ascites buildup. So bringing this to the bench again, looking at the whole cell lysates, the picture on the left are the whole cell lysates run through an SDS page looking at their denatured protein profiles here. 1 and 2 again are the B17 vaccine and the parent strain. We can see very similar protein profiles here. And then when we get to the other 3 novel isolates, we can see some deviations on the banding here too, which is kind of interesting. When it appears in trial A, more of the FP isolates lined up a little bit better. So we do see some differences here. We also ran a quick digestion here with proteinase K to take a look at what was protein, what was carbohydrate. We can see the majority that we were running through was protein, as we did get some higher regions of the carbohydrates here. So it was mostly protein in terms of what we were looking at in terms of antigenicity. So when we ran a Western blot on these whole cell lysates, we chose to use, in one instance, TYS or mock vaccinated mock challenge fish. So these were fish that were, with respect to the trial, naive. They had not been vaccinated. They had not been challenged at the end of the trial. They were a mock group. We can see some light banding from some background here, but not much in terms of what's going on in the Western blot. And now when we looked at some fish that, again, we used some hyperimmunized sera from some hyperimmunized rainbow trout, when we probed the membranes with that from the proteins from the whole cell lysates, we did see some interactive regions, again, in that 250 kilodalton range, and then also some interactions with those novel family Flavobacteriaceae isolates at the same time too, which again we can tie back to and show there's some interactions from a Western blot with respect to cross protection too, pointing that out. So sort of again bolsters what we found with that mixed infection showing protection at the same time too. So just to wrap everything together, what we found from these 2 studies, what we've been keeping at as our research focus, we found that the live attenuated FP vaccine is robust and it protects against a variety of FP isolates as well as some of these mixed family Flavobacteriaceae infections, which is rather interesting too. So what we wanted to move forward with our research agenda here in the interim, as we push this forward, commercializing the vaccine with our industry partner, we want to see if the vaccine provides protection against other emerging bacterial cell-mounted pathogens, of course, too. We'll also look, not just bacterial, but look at some viral pathogens and see if there is any cross-protection conferred when we have an instance of a co-infection too, and then evaluate this vaccine in alternative aquaculture species too. So we've been looking and ran some initial virulence trials in Atlantic salmon as well as brook trout here at U of I, and we're getting ready to look at some vaccination trials in the next few months here with those species, as they do have some potential here as ongoing inland cultured species too. So with that and acknowledgements, I of course want to thank our partners, Tom Locke and Chris at Michigan State University, and then some of our support staff here at U of I, as well as our U of I ARI staff at the same time too. And with that, I'll take any questions that you may have today. And thanks for your time, and thanks to Matt and Nick for organizing this. Greatly appreciated.
Thank you very much. Awesome presentation. You guys have a lot going on there.
Thanks.
So feel free to unmute yourself. We have about 5 minutes here for questions, or you can put it in the chat box and I can read it to Tim.
Hi, Tim. This is Gail.
Hey, Gail.
Beautiful talk. Thank you. It looks so promising. It's really exciting.
Thanks.
The one thing I'm always curious about, that in your second trial in study B, your mixed group, that comes up early, the mortality comes up early, and that it crosses with the mock later. I guess that mixed group, what do you think about the difference between the mixed group versus the individuals? That's a really curious group.
Absolutely, and that opened the door to some questions at the same time too. Is we'd want to probably go back and, you know, we've discussed this, looking at the differences in, of course, fish strains that we had, looking at the 1.5 gram fish from the 11.5 gram fish. But I think this would require some more virulence mapping. And we agree with you, it opens the door as to why that was a faster onset with that infection. And with some of the ongoing RAC project work that we're doing with Esteban's lab, Brent Vuglar is going to be looking into some of those infection dynamics with some of these novel flavobacterial infections too. So that's something I think we need to speculate a little bit more on and discover. Absolutely, I agree with you though.
That's just a classic fish experiment result. There's always one group like that.
Yeah, sure.
Is a little tough, but I appreciate your thoughts. Thanks.
Yeah, no worries.
Hi, Tim. Excellent talk, by the way. This is Hytham from University of Wisconsin.
Hey there.
Hi, how are you?
Hey, great, thanks.
Lots of work, I know, and you know, lots of challenges and vaccinations. So I was wondering, I have one or two questions here. Why the vaccine provided protection against the mix, but not against the single challenge isolates?
Absolutely, that's a great question we've talked about in the lab. Is we're wondering, with the size of fish that we had, you know, using the 7.5 gram fish, if we would have had a little bit more mortality, maybe to work on the dosing a little bit more for that size class, maybe we would have seen a little bit more separation there. You know, it was tough when we didn't see that high mortality.
I agree. I mean, usually when we do challenges like this, you want to hit like 50% mortality at least.
Sure.
To see significant difference when you do a vaccination.
Yes.
The other thing is when I look at the second trial that you did with the larger fish, one of the strains, which one? The T28.
Yes.
It killed 92% of the fish in the first trial, but only 6.7 in the second one.
Yes. Absolutely. That's again something with the dosing. We had worked with Tom and Chris on this, and it can be very difficult. We had some challenges at the start actually quantifying these on our drop plate method. So we went to spread plates at the end, and these emerged very quickly. So I think during this time of the virulence trial, we might have had some variation in the CFU per mil taken, even though the ODs lined up, but the CFU per mil came out a little different. So that's something, again, that I think we're going to work on in the near future here as we investigate more of these isolates coming into UI.
Okay, good luck. Thank you.
Hey, absolutely. Hey, thank you.
Mm-hmm. Hey, Tim. This is Esteban. Great talk, man. And in this same same paper, I have also always, reported, the dose of the challenge based on the amount of CS used per fish. So I am in the same boat. However, recently, due to a suggestion, by Stephen Riley and Matt briefing, and I have seen it, and it does make sense, to start reporting it by gram of fish instead of by total, by basically the total fish. Because
sure.
Particularly in this comparison, you mentioned that that the study B was done in actually larger fish, correct?
Yes.
So I am wondering, if you take into account, I'm not sure if you have, the average weight of, or how much, larger, the fish in the second virulence study was, if the dose actually per gram of fish will actually change a lot the dosing that you were giving to the fish, because injecting 50 microliters is already difficult, to make sure that it went in, but injecting 15 microliters in a 3-6 gram fish, might be even harder. So I don't know if the effect of the mortality is really on those, versus on really damage, made in the muscle also injection. So I'm just curious, you don't have any mortalities in your control, with these guys, or do you guys see any type of mortality as well?
Yes, in the control group, in the post-challenge, not the non-virulent studies, we had a few in the background mortalities, not much at all, but that's a great suggestion as to tying it back to the actual fish weight. And that's something we usually do in our FP challenges too, based on the sizing, we sort of have a range where it's a 50 microliter versus a, 100 microliter sub-Q injection based on that. That's something I think, moving forward, some of these non-FP challenges, we should definitely work out. That's a great suggestion, Esteban.
All right. Thanks again, Tim. That was an awesome
Thanks,
presentation. Nick. Good discussion, guys.